Lieutenant Agent Dante at your service, sir.
Thank you for coming on such short notice, Secret Agent. Well, I'm sure you've heard the news about Secretary General Xu Wenyu.
Yes, sir. The Secretary General Xu Wenyu is a very nice person. He has helped our country so much. Such a pity that he's going to die soon.
Well, that's actually why I've called you here today. You see, Xu Wenyu is going to die very soon from cancer caused by microplastics and carcinogenic materials which have accumulated in his lungs. And that's why I need you to stop this.
Yes, sir, I heard about that. I guess we can't do anything about it because the cancer is there for the last 40 years.
Well, you're right. There's nothing we can do about it now in the year 2062. That's why I'm sending you back to the year 2023.
Introducing Smoking Kills 2. Back to the past.
Behold, my time machine. This time machine has been developed by the most cutting-edge scientists in the year 2062. I'm sure it'll do you well. Your mission is simple, Agent Dante. You must find the young Xu Wen and deliver these nanomachines to his lungs. These will target the cancer, zapping them with lasers and destroying the cancer.
Right, sir. It sounds easy. But where exactly I will find young Secretary Dr. Xu Wenyu?
Well, that's the thing. In the year 2023, he won't be a doctor yet. In fact, he should be just starting his PhD program in engineering at Washington University in St. Louis. You'll find him there, probably outside the engineering building, smoking somewhere. It should be an easy mission. I expect to see you back in 20 minutes. I want you to take this temporal earpiece. This will allow you to communicate with me back in the past.
Great, sir.
Are you ready?
Yes, sir.
Call me when you get there.
Hello, boss. Boss, this is Dante. I have some bad news. I've spilled the nanoliquid all over the ground. Now how am I going to save Secretary General's lungs?
Good news and bad news, Dante. Bad news is you're stuck in the year 2023 for now. The good news is I think there's still a way you can save the Secretary General's lungs using the technology of 2023. But it's going to involve some genetic engineering. Are you ready?
Yes, I can do that, boss.
Great. Let me introduce you to our custom AI trained on the historical scientific methods of 2023.
Hello, I'm Chet EECE. It is a pleasure to meet you, Agent Dante. I'll be walking you through the process for engineering a bacterial strain for incorporation into the Secretary General's laws. First, please submit a lottery ticket for tomorrow's Mega Millions lottery. It is worth approximately $150 million after taxes. The winning numbers are 1, 21, 25, 27, and 40. Next, we have analyzed the Secretary General's laws to contain large amounts of polyethylene, benzopyrene, and vinyl chloride. In the year 2023, these toxins have been accumulating in the lungs of people globally, not just the Secretary General. These microplastics and carcinogens have found their way into lung tissues both directly from aerosols produced by combustion processes as well as through the blood from digested food. Step 1 to solve this issue is to find the enzyme that can degrade these chemicals. Each enzyme is usually specific to just one chemical, so you will need to introduce at least 3 enzyme genes to destroy these chemicals. One such enzyme is ESTA hydrolase, a class of esterase enzyme found in the gut and saliva of the larval stage of the graded wax moth. These enzymes will help break down the microplastics while also providing food to the moth. Breaking down the carcinogens follows a similar methodology. First, we identify the toxin, then we identify a bacterial enzyme which can degrade it, then find a species that produces it from their DNA. For benzopyrene, we could use PAH dioxygenase from Mycobacterium vanillini, and for vinyl chloride, we could use ethene monooxygenase from Mycobacterium aurum. Since this enzyme is a protein, the next step is to find the amino acid sequence for this enzyme. The amino acid sequence can be converted to the nucleotide sequence or the gene sequence that express this enzyme.
Okay, sounds great. Then what should I do with this gene sequence?
You will need to insert this gene sequence into the chromosomal DNA of Streptococcus pneumoniae. You've probably heard of this species as it can cause immunocompromised patients, but is actually present in the lungs of everyone, even when they are healthy, and is quite harmless most of the time. This species is highly adapted to persisting in the lung microbiome and also benefits from over a century of scientific study, including detailed knowledge of its entire genome.
Chat EECE. I think Agent Dante is asking how exactly will we get the DNA to integrate with the chromosomal DNA of the bacteria?
One of the common synthetic biology techniques for genetic editing in the year 2023 includes CRISPR-mediated gene manipulation.
What's CRISPR? Never heard of it.
Well, currently we have cutting-edge technologies to do genetic editing in nanoseconds, but back in 2023, CRISPR is one of the most efficient methods to perform genetic manipulation. First, you will need to construct a plasmid vector that will express the components of the CRISPR system as well as the guide sequence to tell CRISPR where to cut.
But how exactly does this CRISPR system work?
Well, after you construct the plasmid and transform it into the bacterial cell, this plasmid utilizes the host cell machinery to express the Cas9 protein and the guide RNA, which will form the Cas9 sgRNA complex. The complex will bind to the specific gene sequence and Cas9 will cut the gene at the appropriate location. Also, we need to deliver another recombinant plasmid that contains the gene of interest. That is the gene that will express the enzyme as well as the special recombinant protein. After Cas9 complex clips the DNA, the cell has 2 ways to repair the damage. The lung microbiome will also present some unique challenges in terms of your strain engineering. Although the bacteria we'll be using has been previously adapted for use in the lung microbiome, we can further increase our chance of success by considering the conditions our bacteria will have to live in. The first consideration will be that the tiny hair-like cilia of the lungs help to move contaminants out of the lungs. We will either need to optimize our bacteria to invade and survive in between the cilia hairs or bronchial extracellular matrix, or we will need to find a way to get the bacteria to migrate or reproduce fast enough to outpace the outward movement.
Just how fast would this bacteria need to swim against the current?
According to my calculations, the cilia is able to move the gel about 1 meter every 250 minutes. So if you can engineer a bacteria to swim against that, we should be able to survive in lawns.
Okay, I think I got it, but what else do I need to consider?
The next factor to consider will be that the pH will be slightly lower due to the dissolved carbon dioxide from respiration forming carbonic acid and bicarbonate. The nutrient availability will also be pretty sparse and will come mostly from the muscal coating of the lawns, with some supplement from the air. Luckily, this mucus will still contain typical substrate feedstock components like glucose, salts, and lipids.
And what about the immune response? Won't he also have to deal with that?
You're correct. Luckily, since we have characterized the Secretary General's genetic composition and immune response here at the United Nations Hospital, we should be able to engineer the bacteria to display sequences so that his lungs recognize them as one of his own.
Sounds like a lot of factors to consider to design this bacteria. How am I going to optimize the bacteria for all these variables?
You can use the phage-assisted continuous evolution system designed by Dr. David Liu and colleagues at Harvard. This system uses viruses to continually infect and promote mutagenesis in host bacteria while also providing important genes necessary for the survival of the bacteria. Fresh cells are continuously added to the system and liquid is continuously pumped out. Only those cells which reproduce the fastest are able to outpace the continuous loss in population to outflow. Our system could also use a lawn biofilm simulant to coat the reactor protecting cells that are able to successfully bind to it from the continuous outflow.
Agent Dante, remember my motto: no pain, no gain. You should also tailor the challenge for your mutants by gradually changing the pH, oxygen supply, nutrient availability, and surface biofilm until you reach the most challenging condition these bacteria will face in the field.
Yes, sir, I think I understand. But how are you going to get this engineered bacteria to the lungs?
In the year 2023, many strategies have already been discovered for encapsulating living bacteria in small droplets that can be inhaled directly into the lungs using a spray container or nebulizer. I suggest you use the gelatin xylo oligosaccharide encapsulation method for probiotics developed by Tao Huang and colleagues at Nanchang University.
Great! Can't wait to get back to the year 2062.
Careful, Dante. Even with the best technology of 2023, it will still take time to transform, grow, and optimize the bacteria. Have patience with your bacteria, and I'm sure you will succeed.
Thanks, Chat EECE. I'll let you know when the job is done.
Good luck, Agent Dante. The life of the Secretary General, and perhaps the whole world, now lies in your hands.
So I have prepared this bacterial culture. I just need to find young secretary and make him drink. Hi sir, how are you doing?
Doing well.
Oh my god, your breath stinks. Oh, have you heard of this product? This is the new breath freshener. Try it.
Okay.
Try it. Super good. Super nice. You can keep it for yourself. If it ever runs out, you can just refill it through the water. Sounds good?
Sounds good.
Thank you.
Thank you.
Thank you, sir. Have a good day.
Good job.
Hello, boss. Coming, boss. This is Dante.
Yes, Agent Dante, come in.
Yes, boss. Mission accomplished. Now tell me how you'll get back home.
That's great news, Agent Dante. And I've just received a notification that the Secretary General's lungs are in great condition. To get you home, you're going to have to learn about the science of time travel next. Luckily, I think I can do that and teach you how to build a new time machine. First, you're going to need to find some flux capacitors.
Yes, let's do that.